Thank you for coming on a Friday evening, and thank you for having me. And it's lovely to actually finally drive north of New Paltz and sort of down winding roads I haven't visited yet. It's been a decade in this area, and there's plenty of places I'm realizing I really have never been to. So this is a great opportunity. So again, thank you. So yeah, I'm actually going to tell you a little bit about this general concept, I think, that's really sort of central to empirical research generally, not even just neuroscience or anything biomedical in nature, but just to the process of learning and to the process, the scientific process. We think about the process as being one where we design experiments and we have these structured observations, and with those we hopefully answer questions to these hypotheses that ideally also need to be falsifiable. And oftentimes I think people sort of don't think about the very starting point, which is, well, what is the question that you're answering? And if you're asking the right question, then maybe the data that you generate will allow you to learn something of interest. And if you're asking an incorrect question, then all the data you generate will still not provide you information that will be helpful in the end. And so I sort of want to, through two examples in two very separate fields, give you a little bit of a walkthrough of how asking that right question really can make a big impact. And not asking the right question can send even the smartest researchers in the world down the rabbit holes to essentially useless data. And hopefully, again, it won't be a sort of a negative tone. It hopefully will give a little bit of sort of a positive ending in terms of that, as most sort of scientists do, we sort of morph our questions as we learn more information. And so the right question always sort of tends to materialize if we sort of spend enough time thinking about it. So I'd like to give a little bit of a personal story in terms of the right question before I get going into sort of the more biomedical focused information. And that is the experience that I had with sort of coming on to a right question moment. And I was a young graduate student. In my second year, I had just chosen a lab. And for the first year, we sort of rotate through labs and do a couple of different 3-month stints in various sort of mentors' areas of research. And by the second year, we settle onto the lab where we will, in all probability, complete our doctoral research. And so after my second year, so I had spent about a year of work in this one particular lab, working very, very much on sort of a new area for me, which was trying to understand the impact of a specific genetic mutation. And now this was 2004. So the 90s were sort of a genetic revolution, and started the creation of all these genetically modified mice and rats that could be used to actually ask questions about the role of certain genes in conditions that affect humans. And so 2004 was really sort of when this sort of era of genetic research was starting to ramp up. And I was working on one particular gene, and that had been mutated in a mouse model to try to mimic sort of what was happening in humans. And this particular gene was associated with a condition called Fragile X syndrome. It's actually the single largest single gene cause of autism as well. It only accounts for about up to 5% of autism cases, but the overlap is actually the largest one for a single gene mutation. And so here I am, second year of graduate work, and I'm thinking I'm doing this great work. It's exciting, and we're getting interesting data. And as sort of at the sort of the early stages of any field, things are getting tightened, and approaches are getting regulated across different areas of the discipline. And as such, also the way mice were generated in labs was getting sort of more regulated in terms of how we breed to obtain subjects, to test them, to really make sure that any observations are different because of the genetics and not because of some other factors in the lab. And so my lab decided to switch to this new, now considered more appropriate way of breeding mice. And all of my experimental data went poof, flat, all differences disappeared, there was nothing. And I thought, oh God, did I just choose the wrong lab? And did I just commit to do my thesis work in something that was a figment of my mentor's imagination? It's just an artifact, panic attack, went on vacation, I was like, I can't deal with this. And I thought, okay, I'm just gonna deal with this when I get back. And while I was on vacation and caught up on some sleep, I had a thought. The right question popped into my head. And that was, why did my effects go away? What exactly now caused my control group to look just like my genetically modified group? Not that there was no difference, but why wasn't there a difference? And I looked and I compared my old data with my new data. And I'm not going to go into the details, but that led to multiple grants and my entire dissertation and even work that I do now, which was, I realized that by asking why my controls looked like my experimental animals led to the discovery that in the new group, the controls looked like they had the mutation, even though they didn't, which meant that their development was programmed by the mutation in the parental generation. And so, what we refer to as an intermediate phenotype, so an intermediate set of behaviors that were attributed to a genetic mutation had no direct actual relationship to the mutation. And so asking the right question obviously was a big relief to this graduate student back in the day. It allowed me to then continue in that lab, and I had a very successful and happy graduate experience. But it also is an example of how sometimes what could have ended up on the heap of just discarded negative data, that's something I guess I wasted my time on, just by slightly thinking about that question differently, led to actually something quite, it was for me very exciting, right? And I think for my mentor as well, as I said, he got a couple of grants out of it, so I'm sure he was happy. So in that line, I want to sort of talk a little bit less about my right question, but more sort of the idea of asking these types of right questions and how the questions morph from right to wrong and then back to right questions depending on the evidence that sort of is brought in by the very process of empirical research. And so I don't have too many slides, so I'm going to really sort of talk through. So I also want to say I might start sort of using jargon or flying through information that you want me to slow down and explain a little bit more in detail. And if that happens, please just interrupt me. Don't wait until the end because I'll probably forget what I was saying at the time, or you might forget the question. So just feel free to interrupt, please. The two questions that I want to talk about today, very different spectrum of research. One is a neurodevelopmental question in the field of what might be related to the causes of autism. We'll talk about sort of the urgency and the need to understand what contributes to now, since essentially the 90s, the rising prevalence or the overall diagnoses of this condition in children in the US and globally. I'll also talk about another question on the other side of the spectrum, a neurodegenerative condition, Alzheimer's disease, the leading cause of dementia, again, in the US as well as internationally, globally. And understanding what actually causes it, could really yield therapies that don't just deal with symptom management, but actually might what's called be disease modifying. And this is a very sort of topical comment because there are a few things going on right now, and you might have actually read about them, but FDA approval of some of these new treatments that are truly first ones in decades, but also sort of purportedly get at this mechanism of what we think might be causing Alzheimer's, dementia, as opposed to, again, symptom management. So I'll start with my first question on the neurodevelopmental side, and just sort of to get an idea where everyone is in terms of the thimerosal. Who's heard of thimerosal? Anyone? Okay, a few of you. All right, so I'll just kind of back up, and I'm sorry if I'm going to be duplicating information you know already, but thimerosal is actually a preservative that's been used in multi-dose vaccines to inhibit bacterial growth. And it's been used, or it had been used since the 30s or 40s, 1930s, 1940s. And it's a form of mercury, of organic mercury, right? So, yep, that's not good, right? And it's not the same as the stuff that's in fish and that we know has these very deleterious effects on cognitive function and development. But it's a type called ethyl mercury that we know a lot less about. So we know it's not the one that we know is really toxic, but it's an organic mercury. And we don't know a lot of the pharmacology and the toxicology on it. And so even though it's been used as a preservative, it's always been sort of assumed that any sort of safety levels that the FDA would have would be based on just general mercury levels, so the one that we do know something about, right? So thimerosal, again, was this particular component of vaccines, and actually still is to this day of some vaccines, but previously used in most that were what we call multi-dose, so a vial where you would draw out multiple doses to vaccinate multiple individuals. And it really sort of became sort of a topic of huge discussion in the US and globally in the early 2000s. And so autism, sort of autism spectrum disorder, so I refer to it as ASD, which is now the clinical diagnosis, but it's really sort of it's truly a syndrome of various symptoms that can vary in intensity from an individual to an individual, but the crux of it is a social deficit, a dysregulation in social cue perception and output of social communication. It also contains other sort of component core symptoms, and some of which, although not even in the majority of patients, might involve intellectual disability, right? So it's the prevalence, so the number of diagnosed cases in the US and globally have been rising over the last decades. And so the big question is why? And what is contributing to this? So this was, again, in the 2000s, when the numbers seemingly started to increase very drastically. And I'll show you some prevalence curves to sort of illustrate how much sort of the case numbers were increasing over time. That there was a lot of sort of, I almost want to say, frantic searching to see what was happening. Was this something environmental? Was this genetic? What was happening at the time? And so one of the initial points of this potential link that led to this question started in 1997. And the FDA had its, I think it was, I have this written down, it's, Modernization Act of 1997. I actually had to write that one down. But what that act mandated is that the FDA required that all food and medication sort of purveyors and manufacturers of the medication identify and quantify amounts of mercury in their products. So that hadn't happened since 1997, which was also sort of a gasp moment for me. I thought that they were tracking this type of stuff, right? No, not till 1997. And so by the time that that information was made known, 99-ish, right, it became apparent that the thimerosal that was contained in these multi-dose vaccines and that were mandated, right, for children, particularly in terms of school enrollments and particularly public school enrollments, that in a particular dosing strategy that under certain conditions, that some children at 6 months of age would meet or exceed one of the actual dosage limitations set by the FDA for methyl mercury. But as I warned you, ethyl mercury, we don't know much toxicology about ethyl mercury. It's not actually in the environment. It's something that's synthesized and used as a preservative. But it's an organic mercury, right? And so out of a sort of an abundance of precaution, the American Academy of Pediatrics issued a statement with the Public Health Service saying, that's a concern. Maybe we should start phasing thimerosal out, especially of children's vaccines, those that are mandated. The FDA actually followed suit a couple of years later in 2001. And they said, okay, yep, we recommend as well that these be phased out. You don't need thimerosal if you have single-dose vaccines, right? So sort of draw, use, done. And so a lot of the manufacturers basically started shifting to these single-dose. Thimerosal started getting removed from almost all vaccines, and the few that did contain them in children's vaccines, also at least after 2003, and started producing the thimerosal-free vaccines. So why do I bring all this up? Because there was potential for concern. Asking this question wasn't a wrong question. It was an organic mercury compound in vaccines where by sort of putative calculation, certain children following a certain vaccine regimen could reach one of the limits set by the FDA for a similar toxic compound, right? So better be safe than sorry, let's remove it. In addition to this sort of general concern, there was actual correlational data presented to the Institute of Medicine in 2001. This was by a now known anti-vaccine activist, Blaxill, Mark Blaxill, and he presented this data, which again is not incorrect data, essentially showing, and let me see, oh, wonderful. So here in this continuous line, we actually have the prevalence, the number of children diagnosed with autism per 10,000 children. And in the bars, we have the cumulative thimerosal dose based on the vaccinations that the children will be getting if they follow the regular schedule. And most of the increase that we see here from 89 to 93 is the inclusion of the hepatitis B and the Tdap vaccine, which both were the ones that contained thimerosal. For example, some of you might have heard of the study, in the Lancet study 98 that sort of, it's discredited, but linked MMR vaccine, so the mumps, measles, rubella vaccine to autism. Well, that vaccine didn't contain thimerosal, and many others didn't. But the two that became sort of part of the regular schedule probably accounted for that doubling, if not almost quadrupling, of the thimerosal exposure level, anticipated exposure level in children. And so the correlation was presented to the Institute of Medicine as a point of concern to really stimulate sort of further study to look into whether or not there truly was a connection between thimerosal and autism. So the one thing just to go out on a limb and say correlational data is not causational data, right? But with this type of data, there is no way to directly experiment with causational data. And so the best we can do is correlational observational work. But by accumulating correlational and observational studies that all point in the same directionality of effect, they, in totality, can provide pretty strong evidence that something might truly be causally linked, even though it's not a definitive sort of experiment that actually, through manipulating something, shows cause and effect. So if this were true correlation, we would expect to see it repeated from multiple studies using different types of observational techniques to try to get at the relationship. And so immediately after this data was presented, there were studies being published left and right trying to get at that correlational component. And I bring up the other two graphs, and I promise this is the busiest graph data related slide I have, but the data is presented in a similar way. So in the line, we have the cases of autism, slightly different in terms of how they're reported. One is per 100,000 person years, which I don't want to get into, but is an epidemiological method of measuring sort of basically individuals. And then the lower from Denmark was an actual diagnoses of children aged 2 to 10. And what we can see is that over a period from 1980 through 2000, or 98 actually in the lower, we, as these two nations phased out thimerosal for their own reasons, their prevalence or sort of the diagnoses of autism either slowly or drastically increased. And so these type of studies did not match the pattern of the association that was initially presented to the Institute of Medicine here with the California study. And the California study wasn't nationally representative, right? It was sort of a small sort of sampling of individuals in that state, but it was still, again, cause for concern and definitely an appropriate evidence to lead to the question of, is there a relationship, right? But then we start seeing accumulation of other evidence that does not show the same directionality effect. So we can't manipulate, but the observations don't sort of point in a similar direction, suggesting maybe there isn't a relationship. And so basically what I want to show you is that the US, following FDA and American Academy of Pediatrics recommendations, manufacturers removed thimerosal from all vaccines that are part of the infant vaccine schedule. By 2003, early 2003, any stocks of those still containing thimerosal expired. So we can presume that anyone vaccinated after 2003 no longer was exposed to thimerosal levels at any level of concern that was initially detected or measured in that 1999 report from the FDA. But what you can see, and I have illustrated here in the table, and then I just sort of quickly graphed it here, is the actual case. So this is diagnoses per 1,000 children. Average across 11 to 14 sites in the US. They're called the ADAM network, or so the Autism and Developmental Disability Monitoring Network. And we see that the rate overall, also here shown as a 1 in some number, the diagnoses continued rising in the United States. So like Sweden, like Denmark, looking across 11 to 14 different states and sites in those states in the US, also failed to follow up to the directionality of that initial correlational data. And so basically what we have is an obvious increase in diagnoses, with now one of these purported environmental factors that could have been a putative, an element of interest, a putative cause, essentially sort of the data just wasn't panning out. Right? It wasn't showing this consistency of multiple studies going in the same direction. Because again, we can't experimentally manipulate thimerosal. We can't give it to some children and not to some children. That would just, again, not fly for a number of reasons. And so with this data, and then I actually have an image of sort of to exemplify just how much work started being done on, I'm searching just by these two key terms, autism and vaccines. And what I have plotted here in the histogram is the number of published studies that are, and this is PubMed website, so this is Medline database, so biomedical research in the United States, and they also include for anything that is published on the particular topic in peer-reviewed and sometimes not, well, peer-reviewed journals. But what we see is that prior to 1998, there was very little being done on the question of autism and vaccines and the relationship between the two. And the reason for that was that in 1998, in addition to the general sort of information coming out about thimerosal being a preservative in these vaccines, the 98 Lancet study, the discredited study linking MMR vaccine to autism came out, and the 28 total publications that year that used vaccines and autism as keywords, most of them more commentaries and responses to that original Lancet study. But what we see later on is this burst of activity. And in 2004, there were 76 total studies published. So overall, there were a few hundred done by the time that in 2004, the Institute of Medicine, the Immunization Safety Review Committee, surmised that there has been sufficient study over the course of those few years to determine that the body of epidemiological evidence, as I quote from their report, favors rejection of a causal relationship between thimerosal and autism. And at that point in time, in 2004, they deemed that question answered. It wasn't direct experimentation, but the preponderance of observational and correlational evidence suggested that there was no consistent relationship between that particular preservative in vaccines and the rising cases of autism in both here in the US and globally. And so the reason why I bring this up is that at the point when we get an answer to this question, that at this point has been a good question, right? It was a valid question. It was a concern, and it was a potential target, a potential cause, or at least a contributor and a risk factor for autism. At the point when we assess that preponderance of evidence and determine that we have answered that question, then continued asking of the same question, right, becomes something other than scientific inquiry, right? It becomes it's morphed into a financial and time drain of researchers, of finances, right? So the NIH budget, at least this year, is $47 billion, of which 300 plus, a little bit over, million goes or went or has been earmarked for autism research. So it's a tiny fraction of the federal NIH research budget. So in previous years, so in the last decade or so, that budget has sort of gone between 200 to 300 million, which sounds like a lot, but we're talking across all research happening that research in the US by institutions, both actually for-profit as well as academic institutions that can apply for these federal grants. 300 is not a lot. And so when we look at the continuation of publications as an indirect indicator of the ongoing research for autism and vaccines, right, which in 2004 was at the basis of the IOM review, an answered question. I would argue that anything past that point is now becoming financially and research-wise irresponsible, because finances are finite. And funding, and even actually the IOM, this particular report said that money can be oriented and better used to be sort of invested in more promising areas of research. And so because there's a continuation now in pursuing this question, which now morphed from the right question to just something that is no longer scientific, we are not investing as much as possible in areas that could be more fruitful. Not necessarily they would be, but they might actually be the right question now as the next step. And so scientific inquiry morphs. We get new questions based on new evidence, and as that sort of rolls forward, questions that used to be the right questions cease being the right questions and other ones become so. And in this case, this did not happen. And so we are still to this day, and you can see this is in 2023, still publishing data on the question of a link between autism and vaccines. And I just don't know what to say about that other than that we should be focusing on other things. Obviously, I'm biased because this is an area of research for me, and I find it, for me, the interaction between the genetic and the environmental causes to be really fruitful area of research. Again, bias, this is my area. But mostly because we know that autism is heritable, ranging between, in terms of monozygotic concordance, meaning the rate that if one monozygotic, so identical twin, is diagnosed with autism, what is the probability, what is the chance that the other twin will be diagnosed? That ranges. So if something is purely genetic, it's inheritable, and so with identical genetic material, if one person has it, the twin will have it. That would be 100% concordance rate. 0% meaning it's not genetic at all, right? Breaking your leg, that's a 0, right? 0% monozygotic concordance rate. Autism ranges, depending on the study, between 40 to 90%. So sometimes it seems very heritable and genetic, and other times less than 50% of the risk is associated with genes. So there's so much more going on that we don't understand that bears our time and money to investigate. Not the old questions. We needed to morph into some new questions. And in this situation, that didn't happen, right? So I don't want to sort of end on that sort of negative note, because most research has moved away. But there is still evidence of this continuing effort and again, a resource drain, both in terms of time and money. And with that, I want to switch to the other question, which, unlike the autism question, I would say has kind of morphed, maybe. The question is it morphing at the right time due to the right reasons? So I'll let you decide as I sort of move to this one. But one of the, so Alzheimer's disease as the leading sort of cause of dementia is a condition, and so actually I think I have a little bit of a background because what I'm going to talk about now is a little bit more molecular in nature, so that's why I want to lay the groundwork. But it was first described in the early 20th century by Dr. Alzheimer in a particular patient, so August Dieter, who presented with what we would now consider sort of the classic symptoms of dementia. We had progressive memory impairment, loss of spatial orientation, paranoid delusions and other sort of symptoms of psychosis. She was institutionalized and passed away within 5 years of symptom onset. And upon autopsy, it was revealed that in her brain there were these massive sort of deposits of what we now call neurofibrillary tangles and amyloid plaques. At the time, they didn't know it was an amyloid plaque. They called them senile plaques. But they were essentially these, so what is illustrated in the schematic, the neurofibrillary tangles are sort of, think of, and these are neurons or brain cells. They, to maintain these sort of very elaborate shapes, they have to have essentially like a scaffolding inside, right? We call it as a cytoskeleton. And the scaffolding itself maintains that structure. But if that scaffolding collapses, you have a mess of, think of New York City scaffolds, right? If those things collapse, it's just a mess of metal and wood, and nothing's getting repaired and no one can use them for the purposes that they were there to begin with. Well, neurofibrillary tangles sort of are a component of cytoskeletal aspects of the cell. And when they no longer maintain their integrity, the inside of the cell gets these tangled sort of masses of cytoskeletal components, and that interferes with nutrient flow through different areas of the cell, communication, these are nerve cells, so the functional integrity of the cell is severely compromised. The plaques are sort of what's called an extracellular, so they're outside of the cells. And pathologically, they're sticky substances that should be cleared out by enzymatic processes, and they're not. And so they form these clumps, and they physically interfere with the space. They can physically damage the cells and induce sort of toxic environments that can actually also lead to the death of the cells immediately around these plaques. So both through loss of functional capacity as well as through loss of structural integrity, and then these plaques on the exterior sort of causing physical damage, as well as sort of cytotoxic damage, there is sort of brain cell death, and that death is thought to then lead to the symptoms of dementia. Right? And so what I just told you is what is called the amyloid cascade hypothesis. So that was the running idea of what actually causes the neuropathology that leads to dementia, and that dementia, as if it's Alzheimer's diagnosis related, has to involve these plaques. Because there's other dementias that don't involve beta amyloid plaques. Well, they're not Alzheimer's disease, right? That's not the diagnostic code. And so here, this idea that these amyloid plaques were the causal component to the cell death and the cell death to the dementia is part of this question. Is it actually causal? Because for the longest time, we couldn't test that question. There's no way to induce plaques, not in humans, first of all, we wouldn't want to do that, but not even in animal models. We go back to that genetic revolution of the 90s. And all of a sudden, we realized we could. And so I'm going to jump to this figure again. This is illustrating publication rate over time. And you'll notice that, again, here I'm using keywords, Alzheimer's disease and beta amyloid. That's it. You'll notice that unlike the 1,500 studies for autism and vaccines, since 1984, slightly longer span, we've had 56,000 studies published on those two keywords, right? A lot more productive area of research. Also, the budget is slightly different. So the NIH budget for Alzheimer's disease this year is $3.7 billion, so that's slightly less than 10% of its overall annual budget, right? So there's a lot of resources being placed to study Alzheimer's, and the reason for that is actually currently over 6 million Americans are diagnosed and living with Alzheimer's, and it's projected, the number's projected to go to over 13 million by 2050, and the cost to the individuals, to the caretakers, to society is astronomical. And there's a sense of urgency with the aging population that this needs to be addressed. Because by understanding the causes, we can develop treatments. And currently, I can't say currently anymore, until a couple of years ago, there were no treatments that were even theoretically disease modifying, that would actually alter the course of this neuropathology that was thought to then cause the dementia. So, but what did actually spark our ability to start asking that question, what is the cause, was in 1987. So again, there was only 34 studies in 1987. There's over 4,000 already this year, but 34 studies, 2 of which were critical and in providing support to this idea of the amyloid sort of cascade hypothesis. One was the discovery of a mutation in a protein called amyloid precursor protein in a family that was displaying what's called familial Alzheimer's, meaning inherited Alzheimer's, meaning it's a dominantly inherited gene, which means a mutation in this gene will induce typically an early onset form of Alzheimer's. And so they were able to sequence the genes and identify that the mutation occurred in this particular protein, from which the beta amyloid that forms those plaques is actually cleaved. So this is the precursor protein to the sticky component in plaques. The other study was that another lab was able to actually clone APP. And when they were able to clone it, they could clone it into a mouse and create a transgenic mouse, a mouse that actually had human APP. And with human APP, and plus a couple of other configurations, they started generating amyloid plaques. And as they aged, and with amyloid plaques, they started exhibiting memory impairments. So they were what we call a model, right? A model of the condition that has construct validity. What we think is the pathology is inducing the mouse equivalent of the symptoms. Right? I mean, we can't say that a mouse has cognitive decline, but we can test it on certain memory tasks, and they would get worse as the plaque load increased. Right? And so all of a sudden we can ask the question, is this causal? And you'll notice right here, there's this burst of activity right here. These are the 2000s and the teens. I mean, the number of studies using various transgenic mouse lines, looking at beta amyloid deposition and removal and this and that at points of age. And oh my gosh, there's so many, again, thousands a year. And everything was pointing to this beta amyloid, again, deposition and their levels. So how much was there would be correlated to the severity of the memory impairments in the mice. All of this finally generated an interest to say, okay, well, if this is causal, then removing it should alleviate or at least prevent cognitive decline or in mice memory impairment. Come in what we call immunotherapeutics. So immunotherapeutics are essentially biologicals, antibodies, that attack, that recognize certain sequences of protein, amino acids in proteins. So every protein is sort of a sequence of amino acids folded in a three-dimensional sort of configuration. And antibodies in our immune systems, if it's a protein that's not of our origin, they'll recognize some part of these amino acids, attach to it, and tell the rest of our immune system in terms of B cells and everything to attack and get rid of this foreign thing. The problem with beta amyloid is, well, our bodies make it. Our immune system is not going to attack it, right? Typically, if our immune system attacks our own stuff, that's an autoimmune condition. That's not a good thing, right? So how do we get the immune system of an organism to attack and clear an endogenous or sort of something that the body makes on its own, a protein? And so one of the ways to do that is to essentially either immunize actively or passively. Active immunization is sort of the vaccines that we kind of talked about. You inject someone with a little portion, a little peptide of the pathogen, and our own internal immune systems get activated, recognize it, and build up a response. Passive immunization is when you get an infusion of the antibodies already pre-made So think in terms of COVID. The COVID vaccines that weren't RNA-based, or sort of the active immunization, even the RNA-based ones, where our immune system learns to respond to that antigen. Passive sort of immunization is some of the now no longer functional infusions for treatment of COVID in susceptible populations to prevent the onset of severe disease. So if someone presented with COVID and they were in a susceptible population, they would potentially get an infusion of antibodies that would neutralize COVID much more quickly than their own immune system could sort of generate a response. So using this sort of immunological principle, which by the way is sort of what we call a modern sort of area of oncology even, is activating your own immune system to get rid of the things that are harmful to you. That idea is what was being generated in the 2000s and being tested in these mice that were sort of creating plaques in their brains and showing these memory impairments. And the data was so exciting because they would immunize these mice and the plaques would disappear. And sometimes there would be no changes in memory impairment. They would just still be as impaired as when the plaques were there. And then people said, well, maybe the damage is already done, so we have to immunize them earlier. And then studies started coming out showing that, well, if you immunize them at the earlier age when the plaque sort of load was lower, you could actually get memory improvement. And there were some studies showing that if you immunized some of these mice before they ever started generating the plaques, so when they were young, you would prevent plaque buildup and prevent memory deficit altogether. So armed with that, clinical trials. And this is sort of the era of what I want to sort of talk about now. And I have two of them listed here, but just to let you know that these are the two that have been currently FDA approved. There is a graveyard of these antibodies that have tested from 2011 through now even in clinical trials that have failed after they failed after they failed. And by failed, I mean individuals were given the immune therapies. For some, plaque load was reduced, no clinical improvement in cognition scores. In others, no change in plaque load or in clinical scores. So it was a touch and go. Initially, people sort of theorized that, well, maybe this is actually because mice are not men. What worked in mice is not going to work in human patients. And so this is now evidence that this beta amyloid is not causal. If we remove it and it doesn't improve cognition, it can't be causal to the dementia. But just like with mice, people started tinkering with how early can they give it. So you'll notice both of these that are approved only for early or mild Alzheimer's. So mild cognitive impairment, which would be preceding sort of a state prior to an actual Alzheimer's diagnosis, or early diagnosis of Alzheimer's. They're not indicated for, particularly for late or advanced, because as evidence showed in the clinical trials, at that stage, removing plaque doesn't do anything. The damage to the tissue is done. Exactly. But by more careful selection of patients, the clinical trials, our ability to detect plaque now even just as or even prior to symptom onset, particularly individuals who carry the genetic mutation. So there's a wonderful network, again, of individuals who have been genotyped and they know they carry the genetic mutation for one of these familial, which is only about 1% of all Alzheimer's cases, but these familial cases. And these individuals are participating in a lot of this early, what's called biomarker research. How can we detect the earliest stages of someone who is likely to develop Alzheimer's? Because what this immunotherapy suggests, the data, is that the earlier we start, the greater the benefits will be. And so again, let me just sort of talk about the FDA trial approvals here. In 2001, an accelerated approval was given for aducanumab. I love the names, but MAB at the end stands for monoclonal antibody. So if you see MAB at the end of any one of these crazy names, it's an immunotherapeutic, a biological. The first one was approved because it showed reduction in plaque load, but it didn't show clinical improvement. And so that was the controversy where the FDA advisory board unanimously suggested that it not be approved. And the FDA went, no, we're gonna give it this sort of accelerated approval. Because maybe you'll do something. And just sort of my own 2 cents on the matter, I think it was also some sense of frustration, because prior to that point, the FDA approved sort of therapies for Alzheimer's hadn't budged for 2 decades. There was nothing new on the market. And everything that was on the market at that point were 4 drugs. 3 that sort of really deal with modifying the general activity of brain cells, so nothing to do with the neurodegenerative process, and the other sort of targeted again a certain neural sort of chemical that neurons use to communicate. All of these were actually quite effective at improving symptoms for a while, and they would delay cognitive decline by some amount of time, but then it would progress at the same rate as untreated individuals. So the hope here was with the theoretical disease-modifying approach, well, maybe, just maybe, there might be some effects that weren't being picked up, right, on clinical trials. The problem is that's just not how the FDA should really approve drugs. So again, controversy, right? But traditional FDA approval has been given for a second one, the Leqembi, which is lecanemab, also a monoclonal antibody, just a few months ago. And this particular drug will actually likely be covered by, for example, insurance companies and Medicare, whereas the first one isn't. It hasn't really been sort of on the market for long, and it hasn't been implemented because no one, well, Medicare said, well, we're not going to cover, by the way, price tag when they started was $50,000 a year. So they're like, well, you show no clinical benefit and you want $50,000 a year, and monthly infusions and MRI scans and all these are really expensive follow-up care. Lecanemab is not that much better. It's twice monthly infusions. The price tag is a mere $26,000 a year. But again, it does show moderate clinical improvement. And I think I actually have the image. This was released by the company that has developed it, Biogen. And what they show here is this is a cognitive, so this is a particular score. There's a couple of different ones that you might see on these axes, but this is looking at cognitive performance. 0 is the starting point, and that anything in the sort of negative direction would be, as they illustrate here, the worsening of cognitive performance in that individual. And what we see in the black line is the placebo arm of the trial, and then in green are the individuals who are actually treated with, I believe this was, oh, they don't show actually the dosage. Whatever, it was some higher dose of their trial. And what they start to see is an attenuation, a slowing down of cognitive decline. It doesn't stop, it doesn't reverse, but it slows down. And so by the time the trial ended, which was at 18 months, what they found was that there was a slowing of approximately 27%. And they, in their calculation in terms of person years added, sort of calculated that's approximately up to a year of sort of delayed cognitive decline. So it would take this individual another year, right, to reach this level, right? And so these were the full results, and this is what the FDA used to basically approve this particular treatment this last July. Now, the reason why I bring this graph up is because in theory, the argument is that these are disease-modifying treatments. They are actually going to slow the rate of cognitive decline because they're modifying the pathology that causes the cognitive decline. And so this is an illustration of what the graph should look like if they completely stop the decline. That's not something we see. If we slow the decline down, which is what we hope we would see, right? And we think we might be seeing, although 18 months I'm not sure is long enough period to see if it truly is slowing down. And then here in red is essentially what all the other disease-modifying therapeutics have been doing up till now, providing a bit of a delay and then a continued rate, equal rate of decline over time, right? And so what we see with these compounds, or with this particular compound, and then the one that's actually currently under review by Eli Lilly that shows even greater clinical efficacy. So this is a 27% slowing down. The other one, this, it's an MAB, I don't know it off the top of my head, is at around 35 to 38%. So that one seems to be more efficacious for reasons that might be due to the trial design, but it's likely that it will also be approved by the end, basically by the end of this year. And so even though these trials are only looking into 18 months, the question will be, do we see this type of an attenuation of the progress, where if we get better and better at detecting the biomarkers that indicate that someone is highly probable for Alzheimer's, if we begin therapy earlier, do we slowly shift from the blue to the green, if at all possible? And that's sort of the excitement here. So that question that we started with, is beta amyloid actually causing the Alzheimer's disease-related dementia, now seems to actually be positive in terms of answering that yes, it does, at least in part, maybe not completely, but in part causing the dementia. And now I think that question has morphed into how early can we detect preclinical or signs that someone is likely to develop dementia, particularly Alzheimer's dementia, so that we may initiate therapy. So now instead of saying, is it causally related, the question has now morphed into how do we detect it so we can start treatment as early as possible? Because we can't give this stuff in drinking water. First of all, 26,000 is a lot. The other issue is it's not risk-free, right? It's an immunotherapy, and one of the most common side effects is actually a site inflammation in the brain. Not necessarily severe adverse effect, but one that has been noted continuously in all of these trials. And so anything that has a sort of a palpable risk associated with it can't just be given to people who aren't at, let's say, high risk of developing Alzheimer's. So now we have to fine-tune that part. But it seems that we have been able to answer the question of, is that causally linked? Seems that at least partially yes. And now we've moved on to, now how do we administer it more effectively? So here's an illustration of a question morphing in a way that's actually really positive and exciting. And we finally got that answer, right? And we asked that right question, and it took decades to get to it, but it was answered more or less. Now we move on to a new question, which is, when do we need to start the treatment to be most efficacious? So it's an exciting time. I've been following immunotherapeutics for a long time, and I always used to say, I don't think this is happening. All through the teens, it was negative trial after negative trial after negative trial. And these are the first ones that are showing some efficacy, and I think the key was designing the trials to start as early as possible. And it seems to be finally getting to a point where there's some indication that it will be efficacious. So fingers crossed, there have to be longer follow-ups to see that this is actually what they illustrate here, a changing rate of cognitive decline. And if that truly holds, then we have finally a disease-modifying therapeutic. Yeah. Mm-hmm.
In the beginning, your slides talked about amyloid and also tangles.
Yes.
I'm not sure I can even phrase the question, but are they separate enough that you might consider attacking the tangles? If that's a matter of structure, is there some way to alter the structure? Has anybody been looking into that?
So the tangles themselves, no, but there is another type of dementia that's called a tauopathy, and the tau deposits are sort of a different entity. They don't contain amyloid, but are a target of some immunotherapeutics. But they would not be used for Alzheimer's by definition, because they were a different type. Yeah. The neurofibrillary tangles themselves, no. As a target, mostly because they're intracellular. So to get to them, if they were to be available to the antibodies, it would imply that the cell has already been damaged. So it would be difficult to actually target and access them. So, and that's one of the reasons why there may never be a full stop to the cognitive decline if the neurofibrillary tangle component isn't addressed. And I think that's why it's a very good question, but not with the current approach, not with these approaches. Yeah. Mm-hmm.
I'd just like to make two observations. I loved your presentation.
Oh, thank you.
Wonderful. in here, there are a lot of people with gray hair, so this is very interesting. You know, you're not going to have your 20-year-olds in here. But I'd like to point out two caveats. And one is that when you say side effects, some of them are really bad.
Absolutely.
Like brain bleeds,
Mm-hmm.
cerebral edema. These
Yep.
are not, you know, you have a
Yes,
indigestion or something like that.
exactly.
That's number one. And the clinical benefit, you know, in a real world sense is rather small.
Yep.
That's, so we have to be careful about that. I mean, it does not undercut the basic premise of the basic science that you're looking at. But, you know, before people go out and think, oh my gosh, you know, the sun has risen, we're golden here, it's not true. The other thing is, there's an old saying in medicine that a drug has the best side effect profile in the interval between when the drug company prints the brochure and when it starts being used.
Yep.
Because once it starts being used, then everything else starts to pop up, because it's essentially a huge trial
Exactly.
population.
Yeah.
So let's not hold our breath yet.
Well, I would say you're right that what's called post-approval monitoring is a big part of this, because there's no trial that can encompass the sample that would be the public that has now access to a FDA-approved treatment, not without cause for concern, obviously. And one of the reasons the cost of the not just the actual what the company wants to charge for it, but the cost is the monitoring, the follow-up of the patient, again, with the MRI scans, because of some of these severe risks of, again, sort of the inflammatory response in the brain, that has to be monitored. And some of the early therapeutics, actually, with active vaccination strategies, led to severe complications in terms of inflammation in the brain. And so almost all of the drugs that are now being either assessed by FDA or in the late stages of sort of clinical trials are what we call the passive immunization, because they just have a, relative to active immunization, a safer profile. As in, they didn't actually induce death in some earlier trials. So again, yes, absolutely within context. Yeah. 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 Yeah.
So what are people looking at in terms of the cause of autism? Our son was born in 1990, so he was 9 when he was in 3rd grade, and he had a smattering of kids that didn't behave, smart, not smart. It just seemed kind of normal to me. 20 years later, his 3rd grade teacher had to retire because she couldn't take it anymore. Because there were so many kids that were ADD, autism, you know, on the chart somewhere, that she could not teach and could not keep a job where she wasn't going to be yelled at, that her kids were not passing the 4th grade tests that are mandatory. Yeah. So are they looking at things like pollution that might have affected the parents and is passed on to the children, or what is?
So there's a lot. Yeah, so there's a lot of epidemiological studies looking at environmental, sort of population level, environmental potential links. There's a lot of genetic research. There's known environmental causes. So there's certain medications that are known to induce, or are not induce. They elevate the risk of autism diagnosis in children. So, for example, an anticonvulsant used, valproic acid, is a known high risk sort of environmental factor for autism. So there are sort of areas that look at both sort of general environmental factors, specific medications. There's a lot of that environmental intersection with genetics, particularly between autism, for example, and schizophrenia, oddly enough. And so that intersection is, for me, of particular interest, but is also being actively studied. The increase in diagnosis is absolutely real. It's tangible. How much of that is awareness, the changing DSM, sort of now 5 diagnostic criteria? There's different papers saying how much of the rises can be accounted for by some of these essentially administrative changes. I'm not satisfied that it's ever 100%. I've not seen studies that would claim that. But just how much could be sort of a real rise and then what is contributing to it, Unfortunately, we are not very far advanced. Genetically, more than close to 300 genetic risk factors have been identified, none of which, other than the FMR1 and the Fragile X mutation, account for more than a fraction of a percent of risk. Environmental factors such as valproic acid, again, have a very clear, sort of quantifiable risk, but it's not in the general population. This is a very particular type of treatment that it's, again, it's very specific and localized, and actually it's even used in labs as an experimental, environmental, sort of pharmacological way to induce autism-like behaviors in model organisms. But pollution, influenza infection during third trimester of pregnancy, socioeconomic factors, stress. I mean, there have been so many studies looking at all these variables, and one way or the other, they do seem to contribute or associate in one way or another, but nothing is as obvious as this leads to a high probability of that.
What about parental drug use, from their teens on until they started having children? Would that have any effect on it?
So the problem with a lot of those studies is they are what we call retrospective. And a lot of times it's difficult to get accurate information when you're asking people about how they behaved, especially during time that maybe they did things they weren't necessarily proud of, or maybe they do want to boast, so then they say they did more than they actually did of something. So those studies are good starting points to maybe start asking different questions, but they're notoriously sort of subject to recollection bias, or again, sort of factors that might influence someone to maybe not either be purposefully honest or just simply not recollect accurately. And so again, those studies in animal models, there have been plenty looking at manipulations, exposures, but in the human setting, it's much more difficult to track down.
So there is a particular way to test to see if someone is autistic, or you're just going by behavior?
Oh, for autism, so diagnostic criteria are purely observational. They are a set of symptoms, core ones, and then an associated ones that must be met in terms of severity. And then current diagnostic criteria also, for example, I don't know if you're familiar, but there used to be a diagnostic term for called Asperger's syndrome. That no longer exists, right? There is no such thing, and no one can be diagnosed with Asperger's syndrome anymore, because the new DSM, the diagnostic criteria, actually have included Asperger's syndrome and a couple of other diagnoses into the ASD, or the autism spectrum disorder. And now the variation in intensity of symptoms is just that, is a rating of intensity of symptom, both core and any additional ones. So that has now changed as well. And I think you could probably maybe speak to that a little bit, but so the changing, so prior to even the latest revision for the DSM-4 revised, autism used to be a very distinct diagnosis of autism disorder, and there were others, for example, pervasive developmental disorder, not otherwise specified, which was sort of a catch-all sync diagnosis. And as these started disappearing and things started being included into the diagnostic criteria under autism, those numbers, those are what I call the administrative sort of causes that contribute to the elevated numbers. But it doesn't explain the entire growth curve that we see in how many children are diagnosed on a yearly basis. So there's something else definitely going on. What that is, though, is an area of intense study. But again, the consensus now is that the heritable component is likely polygenic, meaning multiple genes. There's not a single gene that would cause or not cause it. The seeming sort of convergence point of a lot of these genes are the way neural cells communicate with each other, and the dysregulation in the communication between these cells. Then, depending on where it happens in the brain region and when in development, could explain then the diversity of symptoms and the severity of symptoms that sort of comprise the ASD diagnosis. But again, these are all very much based on preclinical animal models, because we obviously can't sort of go look at sort of neural connectivity in human patients. And so until we have something that is a testable hypothesis in humans. At best we can do is just try to understand some of these mechanisms in non-human organisms and then apply what we learn in some testable way to patients. But we're not there yet. We're not there yet at all.
To me, it seems like there's more of it showing up now that child care has changed so. You know, that they're, from the time they're 6 months old, they're going into a preschool, whatever kind of nursery setting, and that it's not necessarily going to be noticed as quickly, it's not necessarily going to be handled as well as if they were in a setting at home or with a grandparent where
Right. That's a good, that's a good hypothesis, right? That would be a testable one, looking at early childcare development, care, exposure to certain environments, or failure to expose to certain environments. So those are all testable questions.
it seems like if you're autistic and you're not doing well, and you're now 3, 4, 5, and you're coming home to parents that are tired because they've been working all day, there's other siblings, that there's not really a great deal of hope for improvement until maybe the child is a teenager, or it gets even worse at that point.
Yeah, so clinical practice has gotten better at earlier detection. So what we do see, and this was the latest study by the CDC, showing that relative to current, I know this is a weird way to present this data, but relative to 8-year-olds diagnosed with autism, there are more 4-year-olds currently diagnosed, so meaning that the diagnosis is shifting to earlier and earlier. And what the benefit to an earlier diagnosis is the initiation of the only really known therapeutic strategy, which is applied behavioral therapy. And the earlier children are enrolled and they sort of start working with therapists, and depending on sort of the severity of their symptoms, having different type of access to educational facilities, the earlier that begins, the better the sort of prognostic outlook is for them in terms of living independent lives. Again, depends on the severity, but the access to sort of the system that exists now is based on how early a diagnosis occurs. And there has been a shift, and it has been an earlier and earlier sort of rate of diagnoses in the US, based again on this autism and this sort of the monitoring network, the ADAM network sites. So there is a shift in sort of how early we detect it, and then what do we do? But the question of why does it happen, we don't know. Again, there's all kinds of hypotheses, but nothing really firm.
I mean, one concept also that might be useful to bring up is the idea of a final common pathway of many different insults, which can also
Absolutely.
make it very difficult to tease out. Just because there's a result here, it doesn't mean there aren't many ways to get there.
Exactly, the sort of the convergence point between sometimes environmental, sometimes genetic, the idea that it's not that a specific set of things have to happen, but then there's enough of a sort of a load of risk from a variety of factors converging on a couple of critical systems at critical points of development are sufficient to then elevate the risk and shift, let's say, one child toward developing certain symptoms, and yet some other child that seemingly has the same set of, let's say it's a monozygotic twin, does not, right? But yeah, what those convergence points are, I think, is a more fruitful area of research, because it's sort of a consistency across different manipulations seemingly showing up in tests. But again, most of those are at a preclinical level. And so how those will translate to the clinical setting remains to be seen.
My question is, I read that sometimes parents' fault may develop autism in children if they don't behave properly, if they don't do the right thing, if they treat children incorrectly, and also if they, for example, take drugs like 5-6 years before they will have. Is that effect somehow?
So there have been data showing that in specific instances of parental neglect, and these are relatively extreme, there are lifelong effects on those individuals when they grow up, a lot of times in the sort of mood, anxiety sort of spectrum. Not necessarily autism specifically, but there is a whole host of developmentally sort of regulated sort of brain circuits that are susceptible to sort of relatively extreme neglect. In terms of the one thing that does seem to be associated with autism is parental age, and particularly paternal age, for example. Is that behavioral? Probably not. Maybe the sort of a heritable genetic component that increases risk based on age. But again, it's nothing that's been cleanly identified that is the exact link. So with the parental component to autism, it's actually age is a bigger factor than anything of the others that you mentioned. Whereas for other conditions, sort of anxiety, on the anxiety and mood disorder spectrum, particular sort of maternal parental neglect and actually trauma can be seen sort of in adult individuals later. That has sort of been an associated risk. Any other questions? All right. Well, thank you for coming. Oh, yeah. Sorry.
How much is this research being shared with other countries?
So so that depends on how it's published. So if it is published sort of in the classic sense, there is a 1-year paywall.
Yeah, no, I mean like actively working on it, not the public.
Oh, so actual collaborations,
Yes.
They are very active actually. So especially at the clinical level, recruiting individuals, sites, testing both for disease mechanism as well as treatment strategies. These are oftentimes global. And in the US, a lot of the times it's multi-site because there's no single site that can really carry out sort of the complexities and recruit sufficient number of individuals that are representative of a population.
Excellent. And the second question is, I know there's genetic neutral material that is used to test stuff. Has that been more regulated, or is it questioned, or is it
Genetic neutral.
like the things that you attach DNA to test them? Sorry, I don't know the technical term.
The mutations.
Basically, it's like pre-testing. It's pre-testing. Shoot. It's called.
So like genetic screens for risks,
So
or
there used to be like a standard, like you said, animals, and the animals got changed because of the way they were handled. There used to be a standard for this basis of growing stuff on genetic material.
Like HeLa shells,
Yes.
cells.
okay.
Yeah. So has that changed at all, or is it still sort of a? I mean, I had a friend that actually like sold this stuff to people, which is like so weird.
So that we used to have to clone using cells.
Okay.
But now we can just literally just sequence exact sort of.
Okay, so it's totally reproducible. You're not like gathering stuff from.
So it depends on the research approach, but yes, if you need exact specific replicates of sequence that cannot deviate, you can absolutely do it through DNA RNA sequencing at this point. If you want sometimes, however, to look at the effect of a particular change in a sequence, and then you can actually express it in a particular sort of in vitro or cell model and see what it does there. But the assumption is that as cells replicate, there could be random mutations that occur, and so now you're deviating. You no longer can guarantee, unless you then go back and sequence it, that what you started with is what you're actually measuring at the end.
That's been eliminated.
So it is. There are still reasons to use cell-based approaches, but you
The idea is still.
yes, but you.
sound.
Right. If you need to keep sequence integrity and identical sort of nucleotide sequence, we can do that now.
Yeah, before stuff goes weird, yes.
Yeah, you could get some variability that you couldn't even detect it, much less account for it.
Okay, great.
Yep. Well, thank you again for coming out on Friday evening. Yeah. Good.